Thank you, Mr. Chairman. Ladies and gentlemen, this is my honor and privilege to present my talk to summer school. I will discuss brain injury exacerbates neuropathology of sleep deprivation and neuroprotective effects by nanowired delivery of cerebrolysin and alpha-MSH stimulating hormone. This is a big project supported by Department of Defense, United States of America and Europe of Aerospace Research and Development. I am from Uppsala University and I thank all my collaborators, including Professor Buresanu, for this research and support. I tell you first that all the data and conclusions, they are mine and it has nothing to do with any other organizations I am reporting with. Sleep deprivation is very common in military personnel engaged in combat operations. Even 12-hour or one night sleep deprivation produces serious neurological consequences, and in some of the cases, when these personnel get additional trauma, there is much more exaggeration of brain pathology. So therefore, we need to protect the brain following sleep deprivation and traumatic brain injury. Therefore, we are working on several lines of evidences that suggest that there is a decrease in brain-derived neurotrophic factors and also melanocyte stimulating hormone. You can see from this cartoon that sleep deprivation can influence a variety of physiological parameters, including impaired condition of brain, increase of symptoms of depression, increased risk of breast cancer even, higher levels of anxiety, stroke risk, increased risk for diabetes, weight gain, and heart disease. Actually, sleep deprivation in military is one of the regions where the decision-making situation is affected, and combat stress is very important part of stress. Plus sleep deprivation leads to, this is well known now. This picture shows that the hours of wakefulness, means sleep deprivation, is very similar to that of alcohol-induced impairment of brain function. As you can see, alcohol concentration is increasing, and also the hours of wakefulness from 14 hours to 26 hours, they are very similar in nature. Here you can see that sleep deprivation produces many kinds of changes in brain function, including attenuation of responses to losses following risky decisions. I can tell you here that sleep deprivation is related with alpha-MSH stimulating hormone. Here you can see this is orexin level. Orexin level is increased in obese man, and non-obese man there is a comparison. And here you can see that how it is influencing the alpha-MSH level. Here you can see with the orexin levels. This is an example that shows that MSH protects brain endothelial cells. The experiments is done in cell culture, and you can see that providing additional alpha-MSH stimulating hormone protects endothelial cells. This is just an example, a paper published in Journal of Neurotrauma in 2003 telling alpha-MSH stimulating hormone is decreased in plasma of patients with acute brain injury, and also the alpha-MSH stimulating hormone in critically injured trauma patients. This means that alpha-MSH stimulating hormone is neuroprotective in nature. This graph shows BDNF level in CSF and serum. Both were affected in neurorehabilitation and neurorepair after the brain injury. So here you can see that preliminary association between brain-derived neurotrophic factors, memory impairment, functional cognition, TBI, traumatic brain injury. Thus, what we would like to do in experimental procedures, that we have exogenously supplemented alpha-MSH stimulating hormone, or together with brain-derived neurotrophic factor or different kind of neurotrophic factors, the combination that is called nowadays cerebrolysin. So in present investigation, we have reduced sleep deprivation. After that, we inflicted concussive head injury, that is quite common, inducing mild head trauma, and then supplemented with neurotrophic factors in forms of cerebrolysin, alpha-MSH stimulating hormone. Sleep deprivation was induced in rats using inverted flower pot model. It is well known. And then free movement is also there, but an inverted flower pot is surrounded by water. And when the rats were trying to sleep, they fell down in the water, so their sleep is disturbed. And in those cases, we have concussive head injury after that. This is an example of the model. Here is the rat is suspended on this, surrounding by water. Here is inverted flower pot, and that model is one of the mostly used models for sleep deprivation studies. We have induced mild traumatic head injury by dropping a weight of 114.6 gram from 20 centimeter guide tube over the parietal skull. It produces an impact factor of impact injury of 0.224 Newton. This is called mild to moderate traumatic brain injury. Here I can tell you that some of the nanowired level and nanowired drugs using different kinds of neuroprotection is awarded by National Innovation to my wife Aruna Sharma. Here this is an example that co-administration of nanowired alpha-MSH stimulating hormone cerebrolysin enhances neuroprotection in concussive head injury following sleep deprivation. So this work. Here I tell you something about the methodology. Rats subjected to 48 hours sleep deprivation, and then we have inflicted concussive head injury up to 24 hours. We measured blood-brain barrier permeability by advanced blue albumin and radioiodine, then studied neuropathology. Then we have also measured alpha-MSH stimulating hormone and brain-derived neurotrophic factor levels, and they were significantly reduced. Likewise, the reduction in BDNF levels were also seen in sleep deprivation after head injury. This table shows sleep deprivation induced pathophysiology of brain damage following concussive head injury and their modification with therapy. You can see that advanced blue albumin and radioactive iodine significantly increased after 48 hours of sleep deprivation from 0.3 to 0.89 mean, and here is 0.34 to 1.23. When we combine the sleep deprivation with 24-hour traumatic injury, concussive head injury, the values are further exceeds to 2.34 or 3.24. When we have used alpha-MSH stimulating hormone plus sleep deprivation plus concussive head injury, this nanowired, the values came down to 1.23 and 1.56. When we further added nanowired delivery of cerebrolysin, the values are very significantly reduced to 0.56 and 0.86. Regarding brain edema, you can see that from control level, sleep deprivation alone increased the brain water content from 74.56 to 75.89. Another traumatic injury has further increased to 77.34%. Nanowired alpha-MSH alone reduced the value to 75.23, a little less than sleep-deprived rats. But when we combine with nanowired cerebrolysin, we can see the values is 74.89, very similar to the control level. When we measure plasma MSH level, you can see that in control it is about 20.34 picogram per milliliter. It is decreased after 48 hours of sleep deprivation. Then when we combine, sleep significantly reduced by 8.34 picogram per milliliter. Treatment with nanowired alpha-MSH restored some values 16.34, but you can see when we combine with nanowired cerebrolysin and also alpha-MSH, then the values are restored even higher than the control level, 22.34 picogram per milliliter. Regarding BDNF plasma, picogram per milliliter, the control is 22.34. After sleep deprivation, it is 14.23. Another infliction of trauma to sleep deprived make it to 8.23 only. Alpha-MSH nanowired 17.05 and cerebrolysin and alpha-MSH together made it 23.34, even higher than control group. It means the combined treatment is very much neuroprotective. An example to showing you that sleep deprivation of 12 hour, 24 hour, 48 hour and 72 hour reduces serious damage to the neurons. And this part that we have injected another nanoparticles, it is making worse. When we have used Onden's 5-HT3 receptor antagonist, nanowired, it significantly reduced the cell damage. Here we have intravenous administration of alpha-MSH 100 microgram per kilogram together with cerebrolysin, 5 milliliter per kilogram, 5 milliliter per kilogram. But when we have injected nanowired, then the value becomes even lesser and we can restore the MSH level and BDNF level in plasma. This is an example of head injury. See that right and left sides are different. The injury was made on right side and uninjured side showed much more damage. That is quite similar in clinical cases. When we have combined sleep deprivation and concussive head injury, right side and left side, still left side is showing much more damage than the CHI alone. When we have used nanowired cerebrolysin plus alpha-MSH, it has significantly neuroprotected both left and right hemispheres after sleep deprivation and brain injury. This is also seen at the electron microscopic level. As you can see, this is one cell and left side, and this is one microvessel. We have restored the perivascular edema and the cell is very compact. It means cerebrolysin and alpha-MSH has really protected the brain. This is another example of nanowired alpha-MSH and cerebrolysin 48 hour and after head injury. You can see the treatment has many lively cells that can be seen here as compared to many damaged cells here. So from these observations that I showed you, it appears that TiO2 nanowired administration of alpha-MSH and cerebrolysin induces superior neuroprotective effects following sleep deprivation in concussive head injury. This treatment also reduces alpha-MSH and BDNF in the plasma and brain level following sleep deprivation in concussive head injury. This investigation is supported by various organizations, notably Swedish Foundation for Strategic Research, and at the moment University, NCTR, and all other organizations. I'm not going to take your time. This is also supported by various organizations of this research, and these are my colleagues who helped this investigation. This is a collective effort. And I am from Uppsala University, as I told you. And I thank you very much for your kind attention. Thank you very much, and I look forward to your questions and suggestions. Thank you very much.